Hi, so today we will read about chapter 17, Breathing and Exchange of Gases. As you have read earlier, oxygen is utilized by the organisms indirectly break down nutrient molecules like glucose and to derive energy for performing various activities. Carbon dioxide, which is harmful, is also released during the above catabolic reaction. It is therefore evident that oxygen has to be continuously provided to the cells, and carbon dioxide produced by the cells have to be released out. This process of exchange of oxygen from the atmosphere with the carbon dioxide produced by the cell is called breathing, commonly known as respiration. Place your hand on your chest. You can feel the chest moving up and down. You know that it is due to the breathing. How do we breathe? The respiratory organs and the mechanism of breathing are described in the following section of this chapter. 17.1 Respiratory organs. Mechanism of breathing vary among different groups of animals, depending mainly on their habitats and level of organizations. Lower invertebrates like sponges, coelacanths, and flatworms, etc., exchange carbon dioxide and oxygen by simple diffusion over their entire body surface. Earthworms use their moist cuticle, and insects have a network of tracheate tubes to transport atmospheric air within the body. Special vascularized structures called gills are used by most of the aquatic arthropods and mollusca, where vascularized bags called lungs are used by the terrestrial forms for the exchange of gases. Among vertebrates, fishes use gills, whereas reptiles, birds, and mammals respire through lungs. Amphibians like frogs can respire through their moist skin also. Mammals have a well-developed respiratory system. 17.1.1 Human Respiratory System. We have a pair of external nostrils opening out above the upper lips. It leads to a nasal chamber through the nasal passage. The nasal chamber opens into the pharynx, a portion of which is the common passage for food and air. The pharynx opens through the larynx region into the trachea. Larynx is a cartilaginous box which helps in sound production and hence are called the sound box. During swallowing, glottis can be covered by a thin elastic cartilaginous slab called the epiglottis to prevent the entry of food into the larynx. Trachea is a straight tube extending up to the mid-thoracic cavity which divides at the level of the fifth thoracic vertebra into right and left primary bronchi. Each bronchi undergo repeated division to form the secondary and tertiary bronchi and bronchioles ending up in very thin terminal bronchioles. The trachea, primary and secondary and tertiary bronchi and initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchiole gives rise to a number of very thin, irregular walled and vascularized bag-like structures called alveoli. The branching network of bronchi, bronchioles and alveoli comprises the lungs. We have two lungs which are covered by a double-layered pleura and with a pleural fluid between them. It reduces friction on the lung surface. The outer pleural membrane is in a close contact with the thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The part starting with the external nostril up to the terminal bronchioles constitute the conducting part, whereas the alveoli and their ducts from the respiratory or the exchange part of the respiratory system. The conducting part transport the atmospheric air to the alveoli, clear it from foreign particles, humidifies it, and also brings the air to the body temperature. Exchange part is the site of actual diffusion of oxygen and carbon dioxide between blood and atmospheric air. The lungs are situated into the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by the vertebral column, ventrally by the sternum, laterally by the ribs, and on the lower side by the dome-shaped diaphragm. The anatomical setup of the lungs in thorax is such that any change in the volume of the thoracic cavity will be reflected in the lungs pulmonary cavity. Such an arrangement is essential for breathing, as we cannot directly enter the pulmonary volume. Respiration involves the following steps: breathing or pulmonary ventilation, by which atmospheric air is drawn in and carbon dioxide rich alveolar air is released out. Diffusion of gases across the alveolar membrane. Transport of gases by the blood. Diffusion of carbon dioxide and oxygen between the blood and the tissues. Utilization of oxygen by the cells for the catabolic reaction and resultant release of carbon dioxide, cellular respiration as dealt in chapter 14. 17.2 Mechanism of breathing. Breathing involves two stages: inspiration, during which atmospheric air is drawn in, and expiration, by which the alveolar air is released out. The movement of air into and out of the lung is carried out by creating a pressure gradient between the lungs and the atmosphere. Inspiration can occur if the pressure within the lungs, intrapulmonary pressure, is less than the atmospheric pressure, that is, there is a negative pressure in the lungs with respect to the atmospheric pressure. Similarly, expiration takes place when the intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and its specialized set of muscles, external and internal intercostal between the ribs, help in generation of such gradients. Inspiration is initiated by the contraction of diaphragm, which increases the volume of the thoracic chamber in the anterior-posterior axis. The contraction of the external intercostal muscle lifts up the rib and the sternum, causing an increase in the volume of the thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs, that is inspiration. Relaxation of the diaphragm and the intercostal muscles return the diaphragm and the sternum to their normal positions and reduce the thoracic volume and thereby the pulmonary volume. This leads to an increase in the intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs, that is expiration. We have the ability to increase the strength of the inspiration and expiration with the help of additional muscles in the abdomen. On an average, a healthy human breathes 12 to 16 times per minute. The volume of air involved in breathing movements can be estimated by using a spirometer, which helps in clinical assessment of pulmonary functions. 17.2.1 Respiratory volumes and capacities. Tidal volume. The volume of air inspired or expired during a normal respiration. It is approximately 500 ml, that is, a healthy man can inspire or expire approximately 6,000 to 8,000 ml of air per minute. Inspiratory reserve volume IRV. Additional volume of air a person can inspire by a forcible inspiration. This averages 2,500 to 3,000 ml. Expiratory reserve volume ERV. Additional volume of air a person can expire by a forcible expiration. This averages 1000 ml to 1100 ml. Residual volume. Volume of air remaining in lungs even after a forcible expiration. This averages 1100 ml to 1200 ml. By adding up a few respiratory volumes described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. Inspiratory capacity. Total volume of air a person can inspire after a normal expiration. This includes tidal volume and inspiratory reserve volume TV plus IRV. Expiratory capacity EC. Total volume of air a person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume TV plus ERV. Functional residual capacity FRC. Volume of air that will remain in the lungs after a normal expiration. This includes ERV plus RV. Vital capacity. The maximum volume of air a person can breathe in after a forced expiration. This includes ERV, TV, and IRV, or the maximum volume of air a person can breathe out after a forced inspiration. Total lung capacity. Total volume of air accommodated in the lungs at the end of a forced inspiration. This includes RV, ERV, TV, and IRV, or the vital capacity plus residual volume. 17.3 Exchange of gases. Alveoli are the primary site of exchange of gases. Exchange of gases also occur between blood and tissues. Oxygen and carbon dioxide are exchanged in these sites by simple diffusion, mainly based on pressure or concentration gradient. Solubility of gases as well as the thickness of the membranes involved in diffusion are also some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure and is represented as PO2 for oxygen and PCO2 for the carbon dioxide. Partial pressure of these two gases in the atmospheric air and the two sites of diffusion are given in table 17.1 and in figure 17.3. The data given in the table clearly indicates a concentration gradient for oxygen from the alveoli to blood and blood to tissues. Similarly, A gradient is present for carbon dioxide in the opposite gradient, that is from tissue to the blood and blood to the alveoli. As the solubility of carbon dioxide is 20 to 25 times higher than that of oxygen, the amount of carbon dioxide that can be diffused through the diffusion membrane per unit difference in partial pressure is much higher compared to that of oxygen. The diffusion membrane is made up of three major layers, namely the thin squamous epithelium of alveoli, the endothelium of alveolar capillaries, and the basement substance in between them. However, its total thickness is much less than a millimeter. Therefore, all the factors in our body are favorable for diffusion of oxygen for alveoli to tissues and that of carbon dioxide from tissues to alveoli. 17.4 Transport of gases. Blood is the medium of transport for oxygen and carbon dioxide. About 97% of oxygen is transported by RBCs in the blood. The remaining 3% of carbon dioxide is carried in a dissolved state through the plasma. Nearly 20 to 25% of carbon dioxide is transported by RBC, whereas 70% of it is carried as bicarbonates. About 7% of carbon dioxide is carried in a dissolved state through plasma. 17.4.1 Transport of oxygen. Hemoglobin is a red-colored iron-containing pigment present in the RBC. Oxygen can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of oxygen. Binding of oxygen with hemoglobin is primarily related to partial pressure of oxygen, partial pressure of carbon dioxide, hydrogen ion concentration, and temperature are the other factors which can interfere with this binding. A sigmoid curve is obtained when percentage saturation of hemoglobin with oxygen is plotted against the PO2. This curve is called the oxygen dissociation curve and is highly useful in studying the effect of factors like PCO2, H plus concentration, etc. on binding of oxygen with hemoglobin. In the alveoli where there is a high PO2, low PCO2, lesser H plus concentration and lower temperature, the factors are all favorable for the formation of oxyhemoglobin, whereas in the tissues where low PO2, high PCO2, high H plus concentrations and higher temperature exist, the conditions are favorable for dissociation of oxygen from the oxyhemoglobin. This clearly indicates that oxygen get bound to the hemoglobin in the lung surface and get dissociated at the tissues. Every 100 ml of oxygenated blood can deliver around 5 ml of oxygen to the tissues under normal physiological conditions. 17.4.2 Transport of carbon dioxide. Carbon dioxide is carried by hemoglobin as carbaminohemoglobin, about 20 to 25%. This binding is related to the partial pressure of carbon dioxide. PO2 is major factor which could affect this binding. When PCO2 is high and PO2 is low, as in tissues, more binding of carbon dioxide occurs, whereas when PCO2 is low and PO2 is high, as in alveoli, dissociation of PCO2 into carbaminohemoglobin takes place, that is CO2 is bound to hemoglobin from the tissues is delivered at the alveoli. RBCs contain a very high concentration of enzyme called carbonic anhydrase and minute quantities of same is present in the plasma too. This enzyme facilitates the following reactions in both directions. At the tissue site where partial pressure of carbon dioxide is high due to catabolism, carbon dioxide diffuses in blood, RBCs and plasma and forms HCO3- and H+. At the alveolar site where PCO2 is low, the reaction proceeds in the opposite direction, leading to the formation of carbon dioxide and water. Thus, CO2 trapped as bicarbonate at the tissue level and transported to the alveoli is released out as carbon dioxide. Every 100 ml of deoxygenated blood delivers approximately 4 ml of carbon dioxide to the alveoli. 17.5 Regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demand of the body tissues. This is done by the neutral system, a specialized center present in the medulla region of the brain called the respiratory rhythm center is primarily responsible for this regulation. Another center present in the pons region of the brain called pneumotaxic center can moderate the functions of the respiratory rhythm center. Neural signal from this center can reduce the duration of inspiration and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. To increase in these substances can activate this center, which in turn can signal the rhythm center to make necessary adjustment in the respiratory process by which these substances can be eliminated. Receptors associated with aortic arch and carotid artery also can recognize changes in carbon dioxide and H plus concentration and send necessary signals to the rhythm center for the remedial action. The role of oxygen in the regulation of respiratory rhythm is quite insignificant. Disorders of respiratory system. Asthma is difficulty in breathing causing wheezing due to the inflammation of bronchi and bronchioles. Emphysema is a chronic disorder in which alveolar walls are damaged, due to which respiratory surfaces is decreased. One of the major causes of this is cigarette smoking. Occupational respiratory disorder. In certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope up with the situation. Long exposure can give rise to inflammation leading to fibrosis, proliferation of fibrous tissues, and thus causing serious lung damage. Workers in such industries should wear protective masks. Thank you.